Thank you. So I'm here to talk to you today about Java Clinical Research. We're a clinical research organisation based in Ireland, and we develop customised assays and technologies to help our clients in the pharmaceutical industry find out more about how their drugs work in clinical trials. So just very briefly to introduce the company, we're a Dublin-based CRO, which means we manage clinical trials on behalf of the pharmaceutical industry. So we navigate things like regulatory submissions, identifying sites and investigators, and we monitor and manage studies. And what we also do, which I'm going to focus on today, is we develop customized laboratory procedures to implement in clinical trials, and that can be locally at sites in Ireland, throughout Europe, or throughout the world. So what we do, or how we describe it, is that we, using our assays, we can get better data and more information to clients in the pharmaceutical industry about how your drug is working in a clinical study. So we use modern laboratory techniques to do this, and we have two real ways that we can do that. We can focus on what a drug is doing and measure its effect much better and much earlier than is typically done in a clinical study. Or we can have a much more broad-ranging assay which will give an overview of potential side effects or adverse effects that might not be known or realised, and identify those in early stage clinical studies. So we develop our methods, we streamline them, validate them to industry standards, and along with that we document everything we're doing, and we train sites to either conduct the assays, or we can locate our own scientists in research sites so that we're getting the right sample and processing it correctly, and running the assay either on site, or we can run the assay with a third-party vendor, or we can have them shipped back to ourselves where we will analyse the samples ourselves. So we give an end-to-end coverage of the process. We develop an assay, we implement it, and we can analyse and interpret the results as well. So what all this does, it gives a lot more data, more information to the client, which helps them either sell their drug or their process or their idea, or allows them to make better decisions as they progress through clinical research. So I'll talk through a couple of examples. So one case study was where we were instrumental in showing clinical proof of concept in a first-in-human study. And so here we had a client that had a new drug and had very innovative characteristics, both in terms of how it acted and how it got delivered into the body. They were a small company, a startup, and they wished to demonstrate the unique characteristics of their drug in their phase 1 studies. So what we did for them is we developed 3 customized assays that would show that the drug was getting to where it needed to act, that it was doing what it was supposed to do, and it had the required clinical effects. And we did all this in the first-in-human study. So in a first phase 1 clinical trial, typically you would just be measuring safety parameters and maybe measuring drug levels in the blood. You'd just be getting those 2 very basic readouts of what the drug is doing in that study. So we allowed them to show that actually the compound was doing exactly what it needed to do, that it was getting to where it needed to be, and was having clinical effects in these healthy volunteers. And from that data, they were able to demonstrate that the unique characteristics of this drug, which was their kind of unique selling point, really was there without having to do follow-up clinical studies. So after just running one trial, they were able to sell their compound to one of the large top 10 pharma companies in the world. And this is really critical sometimes for small companies. They may not have the budget to run more than one study, or they might be very limited in what they can do. So by getting us on board, they were able to show a lot more than they would have typically been able to do in their phase 1 studies. So how did we do that? So we have kind of 3 characteristics of how we work that I think helped us in this process. So one is, I would say, agility. So we were able to very quickly assess whether we could do it. And then once we won the contract, we were able to very quickly develop the 3 assays, so they were ready to go in the very brief time frame we had to get everything up and running. And we also had a quick turnaround of results once the study was running itself. We have in-house capability in various areas, and the 3 different assays here kind of demonstrated different aspects of how we work. We had one process where we had a research process that had been developed, and we needed to make sure that would work in a clinical research setting. So it was streamlining the process, documenting it, making sure it was very tightly controlled. In the second one, we developed from scratch a brand new sampling procedure to get the drug target, to get a sample of that so that we could measure drug levels in the clinical study. And the third one was an established assay that they knew would work, but we needed to research the literature to find out exactly what they needed to look at and at what time points to take the samples. So again, customization and validation all in the time frame we had to develop the assays. And the last aspect of what we did is partnership. So we worked very closely with the clinical research organisation, the phase 1 unit that was actually running this clinical study. So they were taking care of all the standard monitoring the patients, the recruitment, the dosing, the kinetics, taking a lot of the standard samples that happen in a phase 1 study. And we added on our customized assays onto that and worked very closely with the phase 1 unit to do that. And that started even before the trial ran, where it was our combined capabilities that kind of convinced the client that we were the right team to undertake this study. So that's a very focused kind of process. The other kind of assay that we can do is much more open. It's a kind of a biomarker discovery type process. And I'll just talk about one example of this is developing a proteomic assay for use in platelets. So proteomics is a way of looking at several hundreds of thousands of potential biomarkers in a sample all at one time. So we have some expertise in proteomics, and we also do a lot of work with platelets, platelet function, and combining these two, we have developed a proteomics assay that gives a very good, kind of comprehensive survey of drug effects without necessarily knowing what a drug is doing. It's very useful for looking for side effects or looking for potential biomarkers of a compound. So we've developed standard methods for sampling and analysing platelets for proteomic analysis. We're recruiting patient groups in various Dublin hospitals to get a good coverage of different types of patients and see what this assay reveals about the proteomic signals that are in their platelets. And we've also done some work with in phase 1 studies and also with some in vitro work where we're treating samples with various different antiplatelet drugs and looking at the patterns that are present in those. And this R&D is being supported by Enterprise Ireland and the Irish Research Council. So the sort of data that this gives is shown here. So we can, these are triplicate measurements of the same sample that vary just in one data point there that you can see where there's a spike of an external protein at different levels. And so we can very accurately measure, very sensitively measure protein levels and look at variations in individual proteins in a very complex mixture. So that's a nice clean sample. And if we look at the overall patterns of protein expression, so really what this sort of process does, it generates a lot of data. When you start to look at a lot of samples, the data organizes itself, or you can look for commonalities in it. And what we have here is 3 different individuals, and when we treat them with different drugs, we can see the same pattern of changed protein expression in those different individuals. So it's a way of showing drug effects. So this is known drugs, and we can also use it to screen for unknown drug effects. So this last example is from a clinical trial where we use this approach. And we have hundreds of proteins measured in lots of different samples, so the samples across the top. And when we look for patterns in there, we see the samples group into three broad categories there. And actually these are samples from people before they start drug treatment, while they're on drug treatment, and after we stop treatment with the drug. And so that's, we found these spontaneous patterns that appeared in the data to show these very subtle drug effects that weren't being picked up by other assays. So we're identifying potential biomarkers that we can then use to look more closely at particular aspects of your drugs. So we can use this in platelets, and platelets are an important drug target, but they're also an important target for drug side effects. So drugs that might cause bleeding or might cause thrombosis will affect platelets in ways that are very difficult to pick up in a clinical study. But we can screen them with this approach to look for potential markers of platelet effects. So these are just some of the people we collaborate on this work. We work with groups in UCD and also DCU actually on laboratory procedures. We work with clinicians in James's and Vincent's to look at patient samples using these assays. And we have some funding from the Irish Research Council and Enterprise Ireland. And I had another slide with all our various clients on it. We've used these different approaches with small and large pharmaceutical companies and also with various other CROs that we work with to collaborate to kind of deliver these assays in clinical studies, whether it's in Ireland, in the UK, or throughout the world. Okay, so that's it. If anyone would like to talk to me about, if you want to find out more about how your drug is working, we can work up how to implement either, as I said, very focused assays or very general assays to find out more about what a drug is doing in clinical trials. Thank you.